Wissenschaft fürs Wohnzimmer.
Einen wunderschönen guten Abend an die Freunde der digitalen Unterhaltung da draußen. Hello, bonsoir, good night. It's really nice to see you all and have you here on our English-speaking episode of Wissenschaft fürs Wohnzimmer. I think it's around 98th episode of this show that is presented to you by the Avis for Future, with this grassroots movement of employees by the Alfred Wegener Institute here in Bremerhaven, Germany. My name is Bjorn. I work in the bioeconomy department on basically trying to make the food for the world from the ocean more sustainable. And before I forget, please, if you like the show, give us a thumbs up. Try to become a follower. And if you have any questions for our guest Cyrille tonight, just put them in the live chat and we will ask those on your behalf at the end of his presentation. As you see, I'm not alone here, but with two of my great colleagues that will shortly present themselves before Cyrille will take over the word.
Hello and bonsoir, everybody. I'm Miriam. I'm a postdoc at AVI as well. I'm working in a different department than Björn, but we are colleagues and working together with Avis for Futures for a couple of years right now. And I'm a marine biologist, but I'm modeling. I'm doing biogeochemical modeling. That means that I'm simulating small algae in the water and look how they are changing with projections of climate change. And now I hand over to Béranger.
Yeah, thank you. So my name is Béranger Colsoul. I am a scientist working at AVI, at Alfred Wegener Institute. I am producing millions of baby oysters on the island of Helgoland to restore the former banks of oysters in the German Bight. So I am half French, half Belgium. So a question in French tonight are welcome. I'm here to help. So Cyrille, the floor is yours.
Hi everyone, guten Abend, ich bin Cyrille, ich bin Französisch und ich arbeite für das französische Institut Ifremer. Ich habe die deutsche Sprache zur Schule gelernt, aber heute ich bin alt und es ist sehr schwer für mich zu sprechen. It's why I switch in English. I'm a marine biologist, and tonight I will present you the first step of space aquaculture. Well, my institute is Ifremer. This institute has several missions: exploration of the deep sea, fisheries, stock assessment, bioresources, and aquaculture. My skills focus on bioresources and fish aquaculture. I'm located in the south of France, close to Montpellier, and you can see on this picture my lab. This is the big roof you have between the Mediterranean Sea and the natural pond. And we have a mission too in this lab. We have a roadmap, and this roadmap is sustainable aquaculture. What happened today? Today we can rear fish in closed system, in tank, and in this system you have an outlet, and this outlet is most of the time solid waste like feces, for example, and you have dissolved waste like nitrogen, form of nitrogen or of phosphorus. What we want to do is to add and completely close the system in the aim to avoid to put molecules into nature and rear different marine organisms like worms, like sea cucumber for the solid waste bioremediation, and for dissolved waste we use microalgae. The idea is also to have a loop for oxygen and CO2 bioremediation, transformation of CO2 in oxygen, and with the algae biomass we can produce a kind of meal, like a fish meal, for example, and we give back this biomass to the fish. The recycling goals in my job today on Earth, it's to have a reflection about water treatment in the aquaculture system, to the molecular recycling, and also to produce more feed, because this system is called integrated multitrophic aquaculture. There are some societal benefits for Earth. And one of them is to increase the food security, because in the same volume of water, before we rear a fish, now we can rear another organism, and you can increase the food security. The second point is the environment and biodiversity preservation. Sorry. You understand that now in this kind of system, there are no molecules in the nature. What happened in 2016? In 2016, the European Space Agency proposed a call of ideas, and they asked to the lab in Europe, if you have a good idea for the future moon base, please tell us. And I had the idea to send my proposition to build a system for fish aquaculture on the future moon base in the moon village. Now, what happened about the food in space? The cost of these two meals, biscuit on the left and on the right, you have a very nice meal, but the cost is 20,000 euro each, because this is the cost to push this meal to the moon, from the Earth to the moon. In addition, if you have a cargo every 4 or 6 months, directly you think about the food autonomy, the partial food autonomy on the moon, because you need to produce something on the moon. You already have some experiment on ISS. You can German people can recognize this astronaut, I suppose. In 2014, there are some experiments with microalgae, photosynthetic organism. The next year, you have the first salad produced on ISS from seed to the salad, but it's always photosynthetic food. That means if you have selected astronaut or moon resident, it's vegetarian people for the moment. And I want to also to focus on the science fiction vision with The Martian. You remember this movie? It's a story of a guy. The story of guy on Mars. He wants to cultivate potatoes. On Earth, we have different vertebrates in cultivation. We read different vertebrates. One of them is the fish, and the fish can sequester and provide in flesh all these compounds of interest, like DHA, EPA, interesting amino acid, PUFA, and B12 vitamin, which is important for the muscle. You know, in space, you lose a lot of muscle. Fish is interesting because it's a champion for the consumption of oxygen. It's the lowest consumer of oxygen. And you know, in biology, 1 mole of oxygen consumed is 1 mole of CO2 consumed. And probably in the moon base, the gas management will be very important, and fish is very interesting for this aspect. Another point is the FCR, what we call it the FCR, and you have a low FCR. FCR is what is how many amount of food you need for 1 kilo of growth, and you can see that the efficiency of feed for the aquatic organism like shrimp or carp or trout, and also marine fish, is very low compared to a beef. A beef needs 8 kilograms of food or feed for a growth of 1 kilo. It's very interesting to have an aquatic organism in space. Another point is the possibility to the fish to participate to emergency surgery in space. You can use, for example, the skin of the fish to fix a burn on the arm. You have a picture here. You can use it because the skin of the fish have interesting collagen and have the good humidity for being a kind of emergency medicine for humans on the moon. And you have also the psychology point. It's very interesting to have fish also on the moon or Mars, because the resident will probably feel themselves like a farmer, like something they were on Earth. And it's very interesting because some publications show that when someone is looking for a public aquarium, you have your heart beating decrease, and your blood also is in good shape. And it's very interesting to have this kind of vision when we are stressed. In fact, fish in space is not completely new. The first fish in space was in 1973. It was an American fish during a mission on Skylab, and most of the fish in low Earth orbit was aquarium fish, very small fish. It wasn't fish for eating after. They were sent for studying the motricity, for example, or the inner hair, what we call otolith. And it was the first fish was in the 70s. In the 90s, a very interesting mission called Seabass. It was a German mission, very, very interesting. This mission inspired me today with a closed loop, with a snail, aquatic snail, with plant, with algae. And I think we can use this knowledge now, today. And last decade, it was more in low orbit, in low Earth orbit. It was more Japanese people, Japanese scientists who sent fish, but always fish, aquarium fish, very small fish. Now, the LUNAR HATCH, sorry, I was trouble with my, okay. The LUNAR HATCH program is the first program with the scientification on what could be a system with fish aquaculture on the moon. Hatching, it's something when the larvae comes, but it's also a kind of window. It's windows on the moon. The question of this project is to answer to, are we able to send fish aquaculture eggs from Earth to the moon and plan the hatching on the European village? Why? Because if we are not able to send this kind of organism to the moon, we don't have to have a huge reflection on what about the aquaculture system on the moon. Okay, why embryo and not juvenile or adult fish? Why? This stage is very autonomous. You don't need to have a feed, for example, in the egg. Everything is in the egg, and we don't need human intervention during the mission. We can also imagine to send fish eggs with an automatic cargo from the Earth to the moon. The metabolism is also very low because it's a small part of the fish. The embryo is very small, and also we need a very small place. Just imagine in the glass you have during the lunchtime or your meal, the glass with water or wine or beer. In this glass, you can have 200 fish embryo. That means probably 200 fish later. It's very interesting because, as you know, it's very expensive to send 1 kilogram in space. The question of the project are, okay, which aquaculture species to send? What kind of module we need for experiment on Earth, on base ground experiment? What is the influence of the launcher vibration? What is the influence of hyper and microgravity on the fish embryogenesis? And also the influence of radiation on the biology of the development of the embryo during the trip from the Earth to the moon. We ask how many fish aquaculture we have around the world. The answer is 360. And on the left, we put all the contrasts of the space mission and all the biological consideration important during this stage. And we succeed to have 20 good candidate for a space mission. Of course, I'm sure that in the question in the chat, you want to say, yes, what are these species? For the moment, I cannot tell you all the species. I will tell you some of them because I want to write a publication later on this and with more information. And with this 20 fish species candidate, we have 7, 8 very, very good candidate. The first project of this program was LOVE, for lunch vibration on fish embryo. And our partner was the Montpellier Space University. This is the protocol, very briefly. The protocol was to put in a device, in a machine, which mimic the Soyuz launch, to put a triplicate of eggs. And we compare this triplicate to another triplicate, brother and sister, very quiet. And we look for, we are studying the hatching rate at the end of the experiment. I have a small movie to show you. I will comment this movie. You can see all the team on it. At the beginning, we have several thousand of eggs, small embryo. We have to select these eggs and put these eggs in a small aquarium inside a CubeSat. You know what is a CubeSat? It's a very small satellite. And the guys control that we put the mimic, the good waves of acoustic waves and on the system. We put probes on it because for the publication we have to show that we use the real vibration of Soyuz. And during the 2 first minutes, this is vibration from the motors of Soyuz. And after the 8 minutes later, this is the waves of the acoustic wave. It's different profiles of waves. You can see eggs in a shadow, and we stop the system, and you can see eggs in the window. And we go back to our laboratory. We remove out the eggs from the aquarium. You can see the device, the machine, and we put them in incubators, in classical incubators, and we are waiting for the hatching. We put here. It's a classical, very classical system. We used to do in experiments, in standard experiment. You can see probably small eggs in it. In few seconds, you have now small larvae. After that, we count the number of larvae, and we are looking if it's good or not, and we can have a result. And this result on the European sea bass, I forgot to tell you that it was this fish, European sea bass. We test 2 stages of embryo, 35 hours after fecundation, fertilization, and 83 hours after fertilization. And you can see that the gray part is the Soyuz simulation, and there are no significant difference between both. That means that this species can be sent in space with a space launcher. We do the same with the migre. Migre is an aquaculture fish from the Mediterranean Sea, and we test it at HPF 14. But this fish is disqualified because its embryogenesis duration is too small. It's only 30 hours between fertilization and hatching. Is disqualified. It's not a good for the mission. Is not good. Now a new project is it's gravity on hyper and microgravity. Hypergravity is the acceleration of the rocket, and we simulate this simulation with a rotor, a very simple rotor like you probably have in your lab. And but this rotor is specific for aquatic organism, and especially is waterproof, in fact. And we simulate from 1G to 5G during 10 minutes. We simulate the acceleration of the rocket. This experiment was performed in the Diese platform at Nancy in France, close to the border of Germany. And concerning the next step, the microgravity, it's always at the Diese platform at Nancy, and we use what we call a random positioning machine. You can see that our aquarium and CubeSat in between 2 arms, and in the center of these two arms, you have microgravity. And we succeed to simulate 26 Earth orbit in this experiment. Okay. The ongoing project concerned this cosmic radiation. The name of this project is CORAF. As you know, radiation come from the outer space, but most of them come from the sun. And in this beam of various particles, you have proton, you have neutron, heavy ions, you have gamma ray, alpha ray, and plenty of kind of particle. And we decide to work on neutron because neutrons are the particle the most dangerous for the biology. Our partner is the French Institute for Radiobiology. This is the experimental room. It's a very huge room. It's a very nice. You can see everything is clean. You have a kind of gun here, and this gun, process neutron. You have a beam with neutron, and you can see here the aquarium, and inside you have a triplicate with in each sample you have 100 embryo, and we also have a sample with the water without embryos because we suspect that radiation can modify the pH of the water. And if you are in the biology and marine biology, you know that the pH is very important, especially in this stage of life of the fish. Okay, in the aquarium. Now, my last video, I want to show you what happened during our experiment, during the experiment of vibration, during the experiment of hypergravity, microgravity, and radiation. This is a nice video, which explains the development of the embryo. You can see that the fish start to be color red. The point, with the point, you have the color in it. At the end, they have no place and no choice to hatch. Now, my last slide. This program, LUNAR HATCH Program, with the idea to send embryo from Earth to the Moon, is in discussion with the European Space Agency to be board on a AL3 mission. AL3 means European Large Logistic Lander. I don't know if you know this project. It's a JAP project, and it's very large lander. And in the payload, you have 1.7 tons of experiment. It's not the weight of the lander. It's only for experiment. You have 1.7 ton. And the first mission for this lander is booked for the geologists and people who is working on ballistic. And the second one will be a biomission, and I'm involved in the definition of what could be this biomission. And only nine experiments are selected for this mission, and we decide to send from the bacteria to the vertebrate all the kind of organism form. You have plants, you have algae, you have worms, insects, and you have a vertebrate, and this vertebrate will be a fish. And we expect that it will be a great mission. For the moment, it's a discussion. It's not sure that LUNAR HATCH can be boarded in this mission, but it seems to be good. It seems to be right. Thank you for your attention, all. And if you have a question, don't hesitate to put the question in the chat. Thank you.
Thank you so much, Cyrille. Well, that was really, I loved it. I'm somehow interested in anyways, all these space missions out there. So I was really curious when Béranger told me about his contact to you, and yeah, you did not let me down. That was really, truly interesting what you just presented us. So thank you for that.
Okay, may I have a last word, please?
Please, Cyrille.
Okay. For the moment, it's a disruptive idea, but I'm almost alone in France, but I'm European. And if people want to join me and probably work on another organism in this kind of mission, we can build a group, an European group, and have several organisms for aquaculture on the moon or Mars or something. It's a call from.
Perfect. Thank you for the invitation to all of you out there that that listen to Cyrille today or any other day that you watch this video. Before we go into any of your questions, let me just shortly tell you about what we're gonna hear in a week's time. That is a week from now. There will be another English-speaking episode on the oceans of the future and the impact of climate change on critical regions. A fellow colleague of ours, Nathan Beach, will talk a bit about how climate modeling works and what are those critical regions and how does one translate into the other and how we predict the future with a special model that is used by AVI also for IPC reports, as far as I know. In addition to that, we have something going on that's the public climate school that is organized, as far as I know, from the students that are former Fridays for Future activists. So they have set up a whole school program, I think, for an entire week that during the day is featuring some typical school lectures. This is all in German. And but there's also an evening program, and just right after the daily news, you can see some of us, including Miriam and two more colleagues, that will talk on the 16th of May. So that this is only in a few days, on how climate models work and what they are actually telling us. So there will be a discussion around that. So if you're interested in that and German speaking, Cyrille, maybe it's good for you also to catch up again with your German. Yeah, feel free to watch that. We will have a link in the description here below in a bit. And yeah, that's it for these upcoming days. And if you want to see me live, I think it must be on Sunday, the 22nd, there's a trade fair on e-mobility in Bremen-Blumenthal, and out there at 12 on Sunday, I will talk a bit about the food for the future. So there you have a possibility to see me live and in color and in person. But enough about that. Let's go back to you, Cyrille, and any questions that we might have in the chat. Miriam, what's going on there? What do you have?
Yes, thanks a lot from my side as well about this wonderful talk. Really, it's a completely new sphere, new topic for me, and it sounds super interesting. We also have a couple of questions in the chat right now, and I start with the first one from Konstantin, and he asked a very specific question, actually. The question is, do the vibrations of a Soyuz rocket differ from a, SpaceX Falcon 9 or Falcon Heavy rocket?
Yes, the question is, what is the difference with the Soyuz vibration and the other rocket? This is the question, right?
Yes.
Okay. Thank you, Konstantin, for this question, because it was our real problem when we have to choose a simulation. And quickly, Abdon belongs to the space community at this time in 2019, and people to the space community told us that if the experiment pass the Soyuz, it can pass all the rocket, because Soyuz do the worst vibration. And I asked to our French astronaut Claudie Henuré, I asked because she used it, and she confirmed that Soyuz is a very shaked rocket. Yeah. Donc it's a good question, because if they pass Soyuz, they can pass the exam for Falcon with no problem.
So it could be that you could find more fish species for other rockets, but these ones are the hardcore species then in the end. Okay, thanks.
Okay, so I follow with the second question from Davy Souza. So the question is...
hello, Davy. I know him.
Regarding the fish food, is it viable to send it within a rocket? there are other alternative ways to produce the fish food in space?
Yes, it's a very good question because it's not sustainable to send water in space, and it's not sustainable to send food for animals in space. And as you probably see on my presentation, my job on Earth is to avoid to use fish meal and fish oil from the fisheries for the feed for aquaculture, and we already have different ways to substitute this with, for example, with insect, with a microalgae, with food waste for the resident, and it's one of the ongoing topic, how to find new sources for feeding fish. And it's very interesting because probably in the greenhouse on the moon, when you eat tomatoes, for example, you don't eat the leaves, but it can be eaten by the fish with no problem. And yes, we have to produce I want to say more than 80% of the formulation of the fish feed on the moon.
Thank you. I have a really interesting question that came back from Portugal, and I was wondering if you then take Portuguese chef to the moon in order to prepare like great grilled sea bass. No, but not for real. My real question would be, and not mine, but Konstantin's real question would be, if you would build fish or bacteria into the walls of the moon station, will they protect the astronauts from the radiation?
Sorry, I don't understand the question.
So Konstantin is asking if you build fish or bacteria, or aquaria, I guess, containing fish and bacteria into the walls of the moon station, if this is, helping them
okay, I understand.
to shield the people inside from radiation from the outside.
Yes, it's a very good question. We already have a reflection about it. If you have humans, you probably have a shield for humans, shielding on the world. But in the addition, the aquatic organism is interesting because it's already in a shield. Water is a matter who can stop the radiation. But probably that the aquaculture system will be in the moon base close to the humans, and the benefit of the shield for the human will be for the fish too. And one of the way for shielding the moon base is to put a thin layer of water around the roof of the moon base. Also, it shows that water is very important, and probably that the fish and aquatic organism will be the best candidate for being better than human, because human is an aquatic organism. You know that the content of water we have is the same than the fish, but we are living in the air, and radiation is more dangerous in the air than in the water.
Thanks a lot. The next question comes from Tao Q, and it's quite interesting for me also as a marine biologist, actually. Have you studied or thought how to assess the impact of space regarding the circadian or tidal rhythm that regulates some critical physiological processes?
Yeah. For the moment, we haven't experimented on it. That's right, because on the moon you have 12 days of light and 12 days of night. And what is interesting is, for example, fish just need to have light for eating. It can be rare and dark. And for example, in industry, when you have an aquaculture fish in a stage of embryo, it's in the dark during the embryogenesis. After, the light is important for the fish because it can see the prey, it can see the feed in the water, and we can imagine to light the tank when we feed the fish. there are different solutions. We can also select a species with less influence, have less problem with the light. You can find in the dark the feed. But we haven't. We know there are some problems, some questions to solve, but I think there are solutions. There are various solutions.
So the next question is an essential question, and I think it's the first question I asked you, Cyrille, when we met, is from Fernando Afonso, is how can we get water, or at least a large quantity of water, on the moon?
Yes, it's a very good question too. We cannot imagine to send water from Earth to the moon, or worse, to Mars. It's impossible. In the next decade, there are a lot of space missions to have a sample of water of the moon. We have water on the moon. There is water. The source of the water comes from the asteroid. You know, the moon have a lot of impact, and sometimes the impact is a kind of big ice from the solar system. And as you probably know, marine biologists know it, half of the water from the Earth come from asteroid. And the moon was also already formed when we have the asteroid shower on the Earth with the water, and probably that the moon also has this water. The problem is the moon have no magnetic field and no atmosphere. And this water go away in space, the molecules go in space. And if we find, we are sure that water exists in the South Pole and North Pole of the moon, but we have to extract this water, and this water will be probably on it's on ice form. That means to convert ice into liquid, it costs energy. And another point, if we don't have water, we don't have human exploration, because we are an aquatic organism. We need the water for drinking and for our activities. And yes, it's completely linked. And we can build also, we know how to do on the Earth. We know that the regolith, the regolith is the dark sand you have on the surface of the moon, and on the regolith you have a lot of hydroxyl. Hydroxyl is OH, the molecule OH, and with two OH you can build H2O2, and you can build easily a water molecule. You have two ways, but this extraction, mining, extraction, and rebuild water, but it costs a lot of energy. And from my opinion, for the human exploration, the key point is the energy. How to export the energy on the moon or on Mars.
Well, clear. So there is an add to the question from Fernando Afonso, is in Earth, we should not drink water from sources we don't know. Like that, how can we convince people that water in moon is safe?
I don't know. I'm a marine biologist. It's a question probably we have to ask. Sure, we have to be sure that this water will be safe. And we don't know at this time, today, we don't know what is this water quality. It is fresh water, it is mineralized water like marine water. We don't know. It's why there are several missions in the decade just for having a sample and study this water. For the moment, we have never touched the moon water. It's very important, because after the analysis of the water, we can choose the aquatic organism for the system.
Very interesting, very interesting. Also, thank you for the questions out there. We try to come to an end. I see, I think, 2 or 3 more questions. The first one I see here is from Gianmarco Dardengo. I hope I pronounced that right. Sorry, Gian. So he says, hello and congrats for the initiative. And this is to you, Cyrille. Do you think that developing genetically modified fish would somehow help to obtain fish more resistant to the new space environment?
Probably yes. For the moment, we test a very basic experiment. It's a disruptive mission, but in fact, it's very simple. And probably yes, because we already know that some fish, the natural selection, the natural genetic selection, because we always speak about the modified organism, but you have a selection also, genetic selection and natural. And we know that, for example, the resistance to the UV, especially in the Pacific Sea, you have different population of fish adapt to UV. You can imagine that probably fish will be stronger if you select a family very resistant to radiation. I think it could be exist because fish was on Earth 3-8 million years before us. And they were on the sea when the atmosphere wasn't in the same shape than now. The atmosphere was how to say, the impact of the radiation. I think in the gene of the fish, I think there are already something very plastic and very resistant for extreme environment, because we are speaking about an extreme environment. If the climate change, for example, change the surface of the Earth, we die, human die, but not the fish, because it can express some gene that it don't use to express and survive. We know that most of the fish from the sea can develop a gene of the adaptation of the salinity. If you have problem of food, of web food in the sea, they can enter in the river and in fresh water and kill all the prey they can find. I think fish is very interesting for that, and it's very good question. In my mind, I think the genetic selection could be a way to select resistant fish, and probably if the guy who asked the question, I don't remember the name, but if you want to work on it, just go. It's open.
Okay, Gian, you heard it. It's up to you now. Go back to Cyrille. You'll find his contact details for sure when you go to the Ifremer website. Look, there's more questions, so maybe one more. Let's see. There's a few questions too. One from Fernando Afonso and one from Walter Quadro-Seifert, and both go towards the water parameters that you can maintain on the moon. So, can we get very stable water parameters on the moon? Is Fernando Afonso's questions, which is, as you said, you probably don't know yet the quality of the water that we'll find there. And Walter is wondering about the nutrient balance of nitrogen and phosphorus. What can be expected of the availability of nitrogen and phosphorus on the moon for fish production?
Yes. There is two questions. The first one is the parameter of for the system, aquaculture system. Our methodology, we change the question when we decide to have a reflection about it. We select fish with high flexibility of the parameter. For example, a fish who can die if you have a variation of 1 degree is not a good candidate. We open the window of the variation of temperature, for example, but also the capability of the fish to resist to a variation of ammonia or something like this. In the 20 good candidates we start to select, there is this parameter. Concerning the temperature, probably it was the question, the temperature will depend of the species, of course, and it's a key because during the night it's minus 150 degrees on the surface, and during the day, the light period, it's plus 150 degrees. If you want to stabilize the water at between 10 and 20 degrees, you spend energy, of course. And it will be also a problem a problem of energy. Everything is a problem of energy. The second question was, I don't remember. It was
the balance of nitrogen
yes.
and phosphorus, how that can be, or how the availability of those two, nitrogen and phosphorus, is expected on the moon for fish production.
Yes, yes. The idea, if you saw my first slide or my second slide, the idea is recycling everything. Of course, on the moon, there are nothing. There are no nitrogen, no phosphorus. That means at the beginning, it will be a very small system, and we recycle nitrogen and phosphorus from, for example, the waste of the food of the resident, of the astronaut, or like I said, from the seeds and the salad or the leaves of the tomato or something like that. We start with this. But in this kind of system, each time you put something new, you can recover and recycle the nitrogen and phosphorus, and more the time will be in front of the adventure, more you will have the opportunity to recycle nitrogen and phosphorus.
Perfect. Very well, Cyrille. This was it. Yeah, this was our last question. I hope I didn't forget any. If so, then please ask them again after the show is over in the comments, and Cyrille will come back to you these days in order to answer them. Thanks to you, Cyrille, on this very interesting presentation. Live long and prosper.
Nice. I expect to come back in 2 years with new information.
I hope so. I hope so. Thanks to you all out there coming to us on a Thursday night and be with us, listening to Cyrille about this really interesting topic. Thanks to you, Béranger, Myriam, for being with me tonight. We close the show as always with our favorite jingle, and I hope to see you next week again in another English-speaking episode when it goes a bit more into climate modeling. Thank you, good night, goodbye, and see you soon.
Bye.
Bye bye.
Au revoir.
Wissenschaft fürs Wohnzimmer.